Welcome to Part 2A of the series on Fescue toxicosis, brought to you by Dr. Karen McDowell at the University of Kentucky's Gluck Equine Research Center.
Thank you for your attention today. Decreased prolactin concentration and vasomodulation, primarily vasoconstriction, are two predominant hallmarks of fescue toxicosis and are the subjects of this presentation. I really believe in species comparisons. We can learn a lot from research that has been done in other species, which is much more extensive than horse research. These are some of the ergot alkaloids of tall fescue that have similar chemical structures to biogenic amines as listed here, and they can bind to biogenic amine receptors and have physiological effects. This is how I've tried to set up these slides. What is in blue are the main points, and what is in black are the finer details. So the main points of this slide are that ergot alkaloids in tall fescue have chemical structure similar to biogenic amines and can bind to biogenic amine receptors. And this is one example. While prolactin is not the only hormone affected by ergot alkaloids, it is probably the best understood. Dopamine is a biogenic amine neurotransmitter synthesized and released by the neurons of the hypothalamus, which is located at the base of the brain. One of the best understood roles of dopamine is that when it is released into the pituitary portal system or bloodstream, it causes chronic inhibition of prolactin from the lactotroph cells of the pituitary gland. Ergovaline is one of the ergot alkaloids produced by common fescue. Dopamine and ergovaline will both bind to the dopamine receptor type D2, listed here as DRD2, on the lactotroph cells of the pituitary gland and suppress the synthesis and release of prolactin. This slide represents just one example of how an ergot alkaloid binding to a biogenic amine receptor results in a downstream effect, in this case the reduction in prolactin concentration. Prolactin is a protein hormone that's about 199 amino acids in length, depending on the species, and we usually think of prolactin as just being synthesized in the pituitary gland, but it is a very versatile hormone that is also synthesized in other tissues, some of which are listed here. Prolactin also affects many tissues of the body, depending on the species, sex, gender, time of the year, stage of the cycle, and many other things. Because prolactin affects hair growth and shedding, its suppression is why some animals maintain a rough, shaggy hair coat well into the summer when they have consumed endophyte-infected common tall fescue. Interestingly, drugs that reduce serum prolactin levels, including ergot alkaloid derivatives, can decrease growth of some mammary tumors. While dopamine serves to suppress prolactin release, these are some of the things that will stimulate prolactin secretion from the pituitary gland. Nursing by the young is a major stimulus to keep prolactin elevated for milk production, hence the name prolactin, or promotes elevated lactogenesis. Effects of ergot alkaloids in cows and ewes, such as fertilization and early embryonic development, or in bulls and rams, such as sperm characteristics, are not nearly this well documented in horses. These are certainly fertile areas of research for equine scientists. Some of the signs of fescue toxicosis may also be enhanced by heat stress. Interestingly, in sheep, fescue shortens gestation length, while in mares it increases gestation length. Ergot alkaloids can also affect digestion. Ruminant digestion is an example of where alkaloids may increase contractility of some blood vessels and decrease contractility of others. Taken together, these changes can reduce blood flow to or from the rumen and affect nutrient absorption, contributing to reduced growth and performance. Interestingly, the right ruminal artery and vein respond differently to ergot alkaloids, and we'll have another example in horses when we get to part 3. This slide shows signs of fescue toxicosis in cattle related to vasoconstriction. Fescue foot, shown on the left, typically in cold climates, is caused by vasoconstriction. It usually begins with swelling and tenderness just above the coronary segment, and areas distal to vasoconstriction may undergo gangrenous necrosis. This usually occurs on the back hoof, hence the name fescue foot, but can also occur in the tail switch and or tips of the ears. Summer slump, again caused by vasoconstriction, is where the cattle cannot dissipate heat appropriately, have unthrifty appearance, rough hair coats that may not shed from the winter due to suppression of prolactin, increased body temperatures, shade-seeking behavior, increased respiration, and reduced milk production. Horses are better at thermoregulation than our cattle, so we don't usually see these severe signs in horses. However, as I said in topic 1, there are some reports of increased sweating, respiration rate, and body temperatures in horses exposed to common fescue, particularly if they are exercising. So how do we scientifically measure vasomodulation? This would either be vasoconstriction, which is the most common, or occasionally relaxation. One way is to measure the size of the vessel with Doppler ultrasonography, which can show blood movement or flow in the vessel as shown in this picture. This is the picture of a caudal artery or tail artery of a heifer. The red circle shows the Doppler signals of the blood flow in the artery. The other way to measure vasomodulation is in vitro or in the laboratory with the use of a myograph. This is from a nice series of studies by Dr. Jimmy Klotz and colleagues at the USDA FAPRO lab. He takes a biopsy of the saphenous vein, as shown in the green circle on the left, and the cattle can do just fine after the biopsy. He then makes cross sections of the vessel to study in the laboratory on an instrument called a myograph. The two pink circles on the right are tracings of the cross section of the saphenous vein, which was used in the myograph. You can see that the vessel on the top has a larger lumen, represented by the black circle. This animal is grazing a pasture with low endophyte content, while the vessel on the bottom has a smaller lumen, and this animal is grazing a pasture with high endophyte content. I had traced the original vessels to make these images. There was no difference in the outside diameters of the vessels, but as you can see, the luminal diameter of the steer with a high endophyte pasture is constricted compared to the lumen of the steer in the low endophyte pasture. This is an example of the graphical output of the data collected from the myograph. As the concentration of ergovaline added to the myograph, shown in the bottom of the graph in blue, is increased, it causes an increase in the contractile response of the vessel, as shown on the left vertical axis in red. Thank you for your attention. Next time I'll talk about Part 2B, ways to mitigate fescue toxicosis, changes in gene expression, and animal genetic variation. Thank you for your time.